Hello, Dr. Barnes. Welcome to my creative response. I will be discussing how catabolism is very cleverly used in the medical field. I couldn't find any cocaine, mostly because I didn't look for any cocaine, and also I don't do cocaine, so sadly, only the topic of this presentation will be on cocaine. As we have learned, glycolysis converts glucose into pyruvate in a relatively simple reaction scheme. Which is very bold of me to say after my performance on this test, but we're not going to talk about that. As we can see, the structure of glucose is changed several different times throughout this process. In 1976, Sokolov, Wolf, and Fowler synthesized fluoro-2-deoxy-D-glucose, which is also known as FDG. This molecule replaces the hydroxy group beta to the oxygen with a fluorine 18 isotope, which is a radioactive isotope. Because fluorine 18 is a radioactive isotope, it allows for the detection of this molecule using a process called positron emission tomography, which is also known as a PET scan. In this synthesis, fluoride ions, typically from potassium fluoride, act as the nucleophile in an SN2 reaction. As you can tell from the mechanism, the hydroxy groups on glucose are all converted into acetate groups in order to minimize side reactions. These acetate groups are then converted back to hydroxy groups using hydrolysis. As alluded to earlier, these FDG molecules are utilized in positron emission tomography. The concept behind this is that antiparticles called positrons are created, and when these positrons interact with an electron, the electron is destroyed and the energy is released as gamma rays. These gamma rays are what is detected by the PET, and areas with high concentration are identified as high areas of metabolic processes due to the high concentration of glucose. This is where the fun begins, as we can finally start talking about cocaine. One particular study compared the whole metabolic activity in the brain of a healthy volunteer versus a cocaine abuser. The researchers found that the cocaine abuser showed significantly less accumulation of FDG in the orbitofrontal cortex uh, than the healthy volunteer, indicating a lower metabolism in that region. This area of the brain plays a large role in cognitive function and decision making. I'm not sure about you, but it seems counterintuitive for cocaine to decrease metabolic activity. So, what gives? Well, from my research, it seems that there isn't one definitive answer to this question. One main contributor to this discovery may be the fact that many cells share common signaling pathways mediated by dopamine. There is an excess of evidence supporting the claim that when cocaine abusers do not intake cocaine, dopamine levels are significantly lower than a healthy person's. This is because cocaine abuse alters the amount of dopamine transporters or receptors. Given the existence of shared pathways and decreased levels of dopamine transporters or receptors, it is not surprising that signaling required to initiate catabolic processes is interrupted, causing this decrease in metabolism. While learning about metabolism one phase at a time, it is easy to forget that all of these processes are connected, and disrupting a system that isn't directly mentioned in class can still affect the system as a whole. It is also interesting to learn how these processes can be manipulated for medical purposes. Um, Here you can find my list of citations utilized for the information presented, and I thank you for your time.